Well, thank you very much for having me here. So I am Fiona Nielsen, CEO of Neurolentech. This is a startup biotech that's spun out of IST Austria. So we are incorporated in Austria, but I'm based in Cambridge, so we're both between Austria and Cambridge. And we are into drug discovery for neurodevelopmental disorders. And I want to tell you a little bit about the challenges of doing drug discovery for diseases of the brain, so you get a little bit idea of the challenges that we're facing. It'll be a very brief presentation, but you're welcome to ask me more questions afterwards. So if you have a disease in the body in some place that's accessible, let's say your skin or even in your liver, you can take a biopsy and you can take it out. And you can have a look at what do those cells look like and what's actually going on in those cells. But what do you do if you have a disease in the brain, or if there's something gone wrong in the brain development, maybe in the early fetal development? You can't just take a biopsy of the brain, or at least I'm sure most of you would immediately say no thank you to this. But how do you then study what's going on in the brain or what's going on in the early development? And when we're talking neurodevelopmental disorders, and probably many of you know this, but we're talking about the kind of disorders that are diagnosed in the early couple of years of life of a child when they're not reaching the developmental milestones, and then they will have a diagnosis based on the symptoms. It could be anywhere from intellectual disability to maybe some motor disorders to maybe epilepsy if they have seizures, or maybe traits of autism. So what we're talking about is how do we look into what's going on in the brain of these children, and how do we identify what's gone wrong that is giving them these symptoms? So historically, the way that this has been studied in biopharma and drug discovery is by looking at mice and rat models. So you actually have, as there's also one mentioned, you already have a mouse model that has been genetically engineered to have a specific mutation. And that has been done for epilepsy, it's been done for autism. But you probably would agree that if you have a rat with autism, it's difficult to say, does it actually have the same level of autism or the same symptoms as a human? Often these things are diagnosed by the delayed language acquisition. So obviously a rat doesn't speak much. So there's a limit to how much you can relate the phenotype of an animal model to what's going on in humans. But the good thing about it is that you have less issues with sacrificing some animals for the sake of science to understand what's going on in their brain, so you can actually have something to look at. Unfortunately, historically, when it comes to neurodevelopmental disorders, there's not been a lot of translatability between the results in animal models and what's going on in the clinical trials when you get to the point of testing on humans. In the later years, in the last 10 years, or maybe even more in the last 5, we've got new technology at our disposal in the labs when we want to look at what's going on in brain diseases and in the central nervous system. We can create what is called induced pluripotent stem cells in the individual. So that means that we can take from blood samples, we can create stem cells which have the capability to be differentiated into different types of cells. So this is quite a discovery. Normally, cells don't go back to stem cells. It's a big breakthrough in science that we're even able to do this. But this means that we now can have stem cells from a patient, that those we can, of course, immediately look at. But even better, we can take these stem cells and we can differentiate them into neurons, the same kind of neurons that are in the patient's brain. So when we create these neurons, different types of neurons, the activating, the inhibiting neurons, and the structural neurons that support them, we actually have cells growing in a dish that are genetically identical and functionally identical to the neurons that are in the patient's brain. And now these neuronal co-cultures, as we call them, we can then study to find out how do the neurons interact with each other, what is the signaling, is the signaling normal, and we can compare them between the cell models of the patient with the cell models of controls, and the controls being either from parents or unaffected siblings, where we can find out exactly what characterizes the disease. The benefit of doing that is that when we do drug discovery, if we can understand why, what is making these cells different, how are they behaving differently, how do they look differently, then we can start making drug screening assays where we can then treat them with a drug and then see if you get back to the normal type of cells after the treatment. And we can, this is just one example. This was in an autism paper where they looked at the expression of genes between the patient models and the controls. And at Neurolentech, we're using not just transcriptomics, oh gosh, not just transcriptomics, but all types of omics data to look at this. So I'll just quickly skip here because I wanted to tell you just very quickly about the work that we're doing with the Tess Foundation. So we have a proof of concept project now where we have collaborating with the Tess Foundation, it's a rare epilepsy with a specific gene mutation, where the Tess Foundation, they have helped us get access to patient samples, and they've done fundraising to help support the research that we're doing, and we are, it's part funded by them, part funded by us. And this setup makes sense because they've given us the commercial rights to the derivatives of those samples so that we have a commercial incentive to pursue drug discovery for that particular type of epilepsy. So it's a long journey. There's many steps you need to go through to get a drug, identify a drug and validate it and get it to market. But at least for the Tess Foundation, we've made it through the first proof points of identifying the underlying pathway that was previously unknown for this particular type of epilepsy. So we're looking to make many more projects of this kind, both with patient foundations, but also with the clinics that we're collaborating with across Europe. So, oops, I'll finish it there. And we're happy to talk to anyone who's interested in collaborating across patient foundations and other biopharma biotech. Thank you very much for listening.